Oh, great. Thank you, thank you, thank you. Okay. So yes, so I'm going to get started today. Today, the topic of our lecture is deep sampling and individual differences in fMRI. My name is Catherine Walsh. Tyler Morgan is going to be doing the second half of the talk, but today I'm going to talk to you about individual differences in fMRI. So I'm going to outline just an overview of this talk. I'm going to keep it simple today. I'm going to talk about what are individual differences, why we care about studying them, and how should we be studying them. So to start out by what are individual differences? I'm going to start by taking a step back and talking about what we think about. Yep. Let's see. Back to the abyss. So starting thinking about traditional fMRI analysis, the kind of things that we've talked about already in this lecture series. So in these kind of analyses, we might take a population of people and have them do a task in the scanner. So maybe I give you either 1 or 3 faces and I have you try to remember them across a short delay and then test you on whether you've seen them before. We have, in this case, we have 2 conditions. We have an easy condition with 1 face and a harder condition with 3 faces. And then we'd use statistics and all sorts of fun math to try to compare these 2 conditions and then get a brain map that will tell us about regions that will care about visual working memory task. But then if we want to think about individual differences, we can put aside these kind of group level analyses of condition. And we can assume that every person has done this task and they get a score. So for this case, the numbers themselves don't actually matter too much, but we can assume that red colors means a person does better, the yellow colors mean a person do worse. We can do a similar thing as we were doing before, compare the easy and the hard conditions, but we can do it for each person. What that does is gets us a brain map for each person. Some of them will look more similar, little bits of differences, but others will look a lot different. We can take these kind of differences in the brain maps, correlate them to performance, and that can tell us something new. So here, for instance, this might tell us that activation in a lateral VFC region is associated with performance on a task. If we take a step back and we think about the variance that we might measure in an fMRI experiment, we can kind of think about breaking this down into 3 pots of variance. First, we can think about between subject variances. So these are the true differences across individuals. So maybe you're just better than me at remembering a face in a task. This is what we talk about when we talk about individual differences. We can also talk about within subject variance. So these are things that might be internal to a person. So things like physiological state, attention, did I have coffee this morning? If I didn't sleep last night because my dog slept on top of me, maybe I'm not going to do as well on the task as I did if I got a full 9 hours of sleep. We can also think about things like experimental conditions. So it might just be harder to remember 3 faces than 1 face. So these are the kind of things we think about when we do group level experiments. To fill out our pie chart, we also have things like we have error, things like physiological or thermal noise in fMRI, measurement error, and behavioral tasks, stuff like that. Most importantly, I do want to highlight that both these kind of between and within subject variance can be interesting targets for prediction, but they are just going to be different kinds of questions. So, well, I'm going to highlight things like between subject variance, individual differences, questions like understanding and how fluctuations in psychiatric symptoms might change, things about developmental trajectories of cognition. These might be more reliant on things like within subject differences. And Tyler is going to talk about this more in relation to deep sampling later in this lecture. So kind of giving you an overview on what individual differences are, let's talk about why we might study them. So I gave you the example of studying a visual working memory task, but that's not the only thing that we can study with this kind of framework. Run Review looked across the literature and found about half of these kinds of individual differences studies looked at different cognitions in general, which we can look at different kind of parts of cognition. Most looked at intelligence, but other tests look at things like attention, reading, working memory, cognitive flexibilities. As specific examples, work within the last decade has tried to predict sustained attention, reading ability, fluid intelligence, and things like personality traits like openness and conscientiousness. If you're paying attention, you'll notice that about 25% of the studies in this review also tried to predict some kind of psychiatric symptom. So we can also think about, within this framework, we can think about psychiatric symptoms as dimensional, not categorical. And these individual differences framework gives us a sense to look at this. So for instance, the NIMH came up with this idea of the research domain criteria, or RDoC. The goal of this framework was to develop, for research purposes, new ways of classifying mental disorders based on dimensions of observable behavior and neurobiological measures. So what that means is we might take people who are diagnosed with some sort of psychiatric illness. So for example, major depression, dysthymia, and bipolar depression, all of which have some kind of similar underlying symptoms, but despite having different diagnoses, putting them all into one big sample, and then trying to understand these more dimensional, continuous measures of symptoms that we can then try to study better. This approach can help us advance our mission at NIMH. We can think about trying to better understand disease pathophysiology. We can try to track disease progression. We can develop new and hopefully more effective treatments, and also try to understand who might respond to what treatments. But wait, are individual differences even meaningful? So a couple of years back, there was this big study that called into question whether individual differences studies were meaningful. They suggested that you need to have thousands and thousands of subjects to have even to reliably say, look at these studies. This finding was picked up by a number of news outlets and generally caused panic in the field. One of the big findings in this paper was that you need a ton of subjects to try to get a reliable correlation between brain and behavior. So to show this, what they did is they took a number of different subsamples of varying sizes from the ABCD study, calculated resting state connectivity, and correlated the connectivity in the default mode network to two different measures of behavior. They showed on one side here the cognitive ability, and the other they showed a measure of psychopathology. What these two panels are showing here is that the correlation, which is on the y-axis, between resting state functional connectivity and the behavioral phenotype varied a lot with the smaller sample sizes, which is plotted on the x-axis here, and eventually converged to a stable mean with enough subjects. They took this further and pulled two specific 25-person sample sizes where they showed drastically different effects within that sample size. One showed a really strong positive correlation, the other showed a strong negative correlation, when in reality, this dotted line is showing that there's really no correlation between these measures. So basically the underlying message was that there are really widely varying effect sizes at small sample sizes, which we often see in these traditional fMRI studies. In response to this paper and these ensuing questions about the validity of these kind of brain-wide association studies, there were a handful of more nuanced and measures papers that came out and said, hey, hold on, maybe let's not panic so much. They did highlight the importance of these big population level studies. Because they do provide, when they're done well, they do provide important information. They look at variance across people. They can be relevant for guiding social policy, but they also highlighted the importance of relatively smaller studies, things that are well powered, these kind of deep sampling approaches with small people and high signal to noise for studying different kinds of questions. I'm going to talk more about these kind of big individual differences studies, and Tyler is going to talk more about the other kinds of deep sampling studies later in this talk. So going back to my third question, how can we robustly study individual differences? So one obvious answer is collecting a lot of subjects. I showed you this figure before, but even that approach has many pros and cons. There are benefits. We get more reliable correlations. We get a wider distribution of phenotypes, and they allow us to use more robust statistical and machine learning methods like cross-validation. But that being said, there are problems. It's expensive, both time, money, people power, getting subjects in. That just expands as you get more people. It's time consuming. And it is also hard to recruit patients. So, not only just healthy volunteers, but oftentimes if you're studying a disease, it can be hard to find people with that disease. So these large N studies are not always feasible to do. So what if I can't recruit thousands of subjects? What can I do? How can I study brain behavioral correlations in a better way? So the first consideration you can make is what brain data you're collecting. Are you collecting task versus rest? Many of the studies that we use in these kind of predictive brain behavior studies look at resting state analysis. We heard a couple of examples in Josh's and Javier's talks earlier in this series. And there are a lot of good reasons we might try to use rest. They're relatively reliable across session. They are kind of trait-like. They reflect functional networks that are a backdrop to anything that happens during a task. So they're kind of an interesting thing to look at. They're relatively easy to measure. There's no task to learn. They require relatively little scan time, just about 15 minutes. And many big open data sets use this. So if you're just looking for data online, a lot of data sets have this. But is it the best for prediction? It might not be. There have been some recent studies that came out that have suggested that if we try to predict fluid intelligence from a number of different scan tasks, so things from the Human Connectome Project or a separate Penn Neuroscience Consortium data set, different kinds of cognitive tasks might predict and explain more variance in fluid intelligence scores than just the rest. You can kind of think about this as like a stress test in doing task versus rest. A task might better drive what's different about us. You can kind of think about it as like a cardiac stress test. Like if you're looking for something wrong in the heart, you're not going to have someone just sitting there. You push your heart, you have them walk on a treadmill to try to bring out what is going on that's different. Other reviews have also suggested that this is not necessarily an either-or decision. You can use more complex models for predicting behavior on a task and individual differences that include both task and rest. If looking at studies that use multiple modalities, so this bar plot up here, tend to do better than just using a single modality model, whether that's task or rest. It's possible that integrating different neuroimaging features can improve prediction performance because they're targeting unique aspects of brain and function, so they better characterize individual differences. Yeah, so that's just what I just said. We can also think about the kind of behavioral measures that we want to use. Thinking about self-report or behavior, just an actual cognitive behavioral task. So let's take an example of face blindness or prosopic. If I want to try to measure how well someone's memory for faces is, on one hand, I can use self-report measures. I can just ask them. I could ask them questions, how do you agree with the fact that my face memory is worse than others? And keep going down the list. Or I can have them do a task to test this. So something like the Cambridge face memory task, where I give you a bunch of faces, I wait a little bit, and I say, hey, is this one of the faces that you just saw? But like I said before, and everything in psychology and neuroscience, these have pros and cons to them. So for example, for self-report measures, they are easy to administer, they are quick. They might be more relevant for psychiatric conditions, asking about symptoms, and they're potentially more stable than an individual. But that being said, there's potential for bias. You are asking someone to judge their own performance. We as people are not always great at that. So it relies the ability to introspect. On the other hand, cognitive tasks might have less potential for bias. It's harder to cheat on a task. It's harder to cheat. If you think you're bad at something, you will do bad on it, whether or not you say you're good at it. These are also things that you can do in the scanner. So if you want to directly measure what your brain is doing when you're doing a face memory task, you can do that with something like the Cambridge face memory task. They also might be easier to predict. A semi-recent paper did show that if we look at trying to predict self-reported measures versus task performance, models tend to do better predicting these kind of cognitive tasks. But that being said, They might have less relevance for biomarkers. So if we're trying to figure out a marker for insert disease here, they might not be quite as good at that. And they are potentially less generalizable. They're really good at testing a single thing, but that might mean they aren't quite as relevant for something else. So now say we've decided we do want to use a cognitive task to look at individual differences in fMRI. We also have to think about what kind of task we want to use. So going back to the beginning of this talk where I broke down individual differences and scores into within and between subject variance, and I reminded you that both kinds of variance can be interesting targets of prediction, but they're just different goals, and that's totally okay. I want to expand on that by highlighting, again, different questions necessitate different tasks. And we're going to do that with a little bit of hand-wavy math here. So if we're thinking about within subject effects, so these are things like group or condition differences, we often think about this in terms of a t-test. And again, not much math here, but we kind of think about that in terms of the difference in conditions on the numerator and the error in the bottom. When we think about this error term, we can break that down into things like the variance between individuals, and so things that we care about in individual differences, but that don't really matter for different conditions, and then just other kinds of error. If we want to measure things like between subjects effects, so things like individual differences, we might use a measure like the interclass, the ICC. So in this case, what we care about is the variance between individual over the error. So say we take a task where we're trying to minimize the variance between individuals because we want to try to maximize the difference in conditions. We then get a bigger t-value. Life is good. We get our statistical significant result. We publish our paper. Great. But if we have a task that's geared to do that, we reduce the variance for individual, we're actually going to also reduce the thing that we care about for individual differences. So the long and short of this kind of hand-wavy math is that tasks that might be optimized for within subjects effects might be poorly optimized for between subjects effects. So thinking about what task you're using and what the goal of it is also important for these kinds of studies. If you want to learn more about it, I have a citation here that includes more math and a little bit more explanation of this. Another thing we also want to think about in terms of choosing our task is maximizing the reliability of our task. A key assumption of thinking about individual differences is that we are measuring reliable trait-like things. Put another way, we're measuring things that are reliable. So I'm going to give you a toy example of what that means. So say I've got this task that I'm administering and it has 6 trials. First trial, great, you get it correct. Second trial, wonderful. Third trial, you mess up, you get it wrong. Not a big deal. We pull it back together for trial 4 and 5, and 6, we get it wrong. Overall, we got a 66% accuracy, life is good. But say we take a subset of trials. Maybe we split out even and odd trials, we calculate accuracy on each of those samples, and we get the same accuracy on both of those subsets. We can shuffle our trials again, do the same, get the same accuracy, and again, and we can do this any number of times. And if a task is internally reliable, we'll get mostly the same accuracy across any number of trial splits. This type of reliability is called split half reliability, and a high split half reliability suggests that a task is internally consistent. So that is, it's measuring the same construct all the way through. A task that has good internal reliability is going to be better to try to measure individual differences than a task with lower reliability. But the big question is, do all tasks just that you're finding wherever have a decent split half reliability, and how can we know about that? So some work I collaborated on in my PhD, we set out to test exactly this. We took a battery of tasks, behavioral tests, split them into subsamples of trials. So for example, even and odd trials, calculated the score across those trials, and then correlated the scores across the subsamples. We did this a ton of different times to create a distribution of scores across subsamples. And then we did this with varying number of trials in those subsamples to get this curve of split half reliability as a function of how many trials went into that reliability. So we did this for a number of tasks. and results varied. So what I'm showing here is that for the Cambridge face memory task, so a task that's widely used in the face memory literature, that we do eventually get a stable mean, that we are able to get a score that is the same across trials. But what was interesting is that the number of trials it took to reach an acceptable level of reliability varied drastically, and it did not always coincide with the number of trials that were actually in the task. So you can see another task where this is a motion labeling task, it doesn't really plateau to a stable level of reliability. For instance, and then in contrast, this Cambridge face memory task converges around this kind of 100-ish trial mark. And even then, it only, if you take a long form of this, it only has 72 trials. So that kind of can be a problem across tasks. But okay, so what? I showed you some math, showed you some permutation tests, great, whatever. But what does that actually mean for me when I want to try to study individual differences? So to try to test this, what we did is we took two tasks that we know should be relatively well correlated. So for instance, the Cambridge car memory task, a very similar task to what I was showing before, but with cars, not faces, and just a visual car matching task. If I show you a car and 3 others, can you tell me which one is the same? These should be pretty strongly correlated. What we did is we subsampled trials from those to get the 2 varying level of reliabilities, took the scores across the 2 trials and correlated them. And then what we showed is that when the tasks were less reliable, so over here on the x-axis, the correlation was lower than with more when the tasks were more reliable because they used more trials in calculating the scores. So even when they were truly related, correlations become lower. And this is mathematically the same for brain behavior, brain. So any kind of these individual differences analyses. But a lot of things that you just want to pause for any questions before I move on. Okay. So I've shown you a bunch of things about optimizing variance in your task, but it's also important to think about what kind of population you're sampling from, particularly if you're looking for behavioral phenotypes that might be associated with psychiatric illness. So for instance, this meta-analysis that I keep referring back to showed that if you're trying to predict psychiatric symptoms, you do a lot better if you actually test a population of people with psychiatric symptoms. And this kind of intuitively makes sense. If you've got healthy volunteers who don't show a lot of variance on whatever phenotype you're trying to predict, your model can't pick up on that variance to actually try to predict well. We can also think about trying to, on the other side, we talked about behavioral tasks, we can also think about our fMRI data. So in this slide that I'm showing some figures from Dan Handwerker, we can see that there is a relationship between the number of subjects, which we have on the y-axis, effect size, and statistical power. For a quick stats primer, for those of you who don't know, power is how sure you are that you can see an effect when it's actually truly there. So let's focus on the blue line here. The general story is going to be the same for the orange line, but let's just focus on one. So say we have an effect that requires, for a given level of sureness about this effect, we need 80 people to show that effect size, to be sure we're going to show that effect size. Doesn't matter what that effect is. It could be the same lateral prefrontal finding we saw before. It could be something else. So if we're able to improve our contrast to noise, which we can say here is like the effect size in our brain, even just 10%, we need fewer subjects. So here we need only about 63 subjects. So while these slides were designed to think about group differences, the same logics follow for between subjects individual differences analysis. If you have a clear effect size, a stronger relationship, you need fewer subjects to show that effect. I'm not going to go too in depth into practical ways to improve your data quality because we've talked about this. Others will talk about this a lot more in other lectures in the series. But just as an overview, you might think about doing like using different acquisition parameters, things like multi-echo acquisitions. You might think about trying to reduce head motion, which if that is correlated with your behavioral measure, can throw big kind of confounds with your data, or trying different pre-processing methods, which might help remove noise from your data. Going to change gears a little bit again. So we've talked about optimizing experimental design, thinking about your task, thinking about your data collection, and now we're going to talk a little bit more about analysis. So the first thing that I'm going to talk about here is cross-validation, and I want to try to give an example, set the stage as to why we might care about this, and that is this concept of overfitting. So imagine you have some kind of variable X and another variable Y, and there is a linear relationship between the two. So when X goes up, so does Y. These could be scores on a car matching test. These could be activation. This could be activation of brain behavior. It could be anything, doesn't really matter. We can take our data and fit a model to it and get really, really specific and add a lot of terms and fit our data really, really well. But then imagine we take a new sample, these red dots, drawn from that same distribution. If we use that really specific model that we used before, it's not going to do well. We fit our model to the noise in our first data set. In contrast, something more simple like a linear model will serve us much better. This all is to say that if we use our whole data set to fit a model and try to understand relationships, we can lead to fit to sample specific characteristics and make you think you're doing better than you actually are. So how do we fix this? One way to do this is a machine learning concept called cross-validation, and I'm going to give you an example of what this looks like. So say we have our sample data. We got 5 subjects here, 5 scores. We can split this into a training and a test set. So what we'll do is we will use these first training set to train the model will make some predictions about our data. Sometimes they're right on it. Sometimes they're a little bit off, whatever, doesn't super matter. We can then take that model and apply it to our test set, which keep in mind, the model has never seen. So it has never seen this data. It will give us a prediction. And then we can compare this actual value to the predicted value to see how well our model is performing. And we can use how well in our test set, our predicted value matches the actual value as a measure for how well our model is actually doing and how well it generalizes to new data. To make sure we really want to generalize our data well, we'll often use what we call k-fold cross-validation. So what this does is takes our data set, so say we have our 6 subjects, we will iteratively break up our data into non-overlapping training and test sets. So what that means is we might first take our samples 3 through 6 as our training set, make our model, test it on 1 and 2. We'll get a score for that. Great, life is good. We'll take our data set again, and this time we'll use a different test set, different training set. So same data, different model, next score. We'll do it again. Another test set, another training set, another score. We can then look across all of these rounds, take an average, get an average over score, and that gives us a better sense about how well our model is doing. And this helps us test these models with relatively fewer data subjects. Now, some actual data to show you how important cross-validation is. One study here took some data from the Human Connectome project and tried to predict behavioral phenotypes with and without using cross-validation. They broke the sample into a discovery sample where they used to train the model, find the relationship, and then a replication sample where they looked to see if that same relationship held when the model hadn't seen. When they didn't use cross-validation, what they saw is that the correlation in the discovery sample is biased to be higher than in the replication sample. So that means this whole cloud of dots, of each of which here is a correlation between predicted and actual performance, is higher on the y-axis here in the discovery sample than it is further out in the replication sample on the x-axis. And this is suggesting that we are overfitting to our initial sample. When they used cross-validation, this bias is eliminated, and there are similar correlations between the behavioral and the brain measure in the replication discovery set. As you can see, that things are more along the identity axis here. Yes, so yeah, so it does. So we're doing better and less biased with cross-validation than without. Cross-validation leverages these held out portions of your data to see whether your models generalize, but a more stringent test of doing this is actually using an entirely independent sample. So one nice example of this is a study from Monica Rosenberg, where they used task and rest data to try to predict scores on a sustained attention task using connective predictive modeling, a method that we've talked about a little bit before in other lectures in the series. And they were able to do so. They could predict well. Life is good. They did this to define a network of connections in the brain that were particularly relevant. Woohoo, great, wonderful, we have a finding. They then turned and this was all trained in healthy volunteers. They then took this model and applied it to an entirely new sample of people with ADHD, so people with issues in attention, and tried to predict symptom severity, and they were able to successfully do so. So the fact that this model that they developed in healthy controls was able to predict ADHD scores in a new independent sample was a great test of how useful and valid it was. So that's what I've got for you today, kind of wrapping up. Just going back to what are individual differences. So they are a statistical association between some sort of brain measure and some sort of behavioral measure. We should care about them for a number of reasons, including giving us new insights into nuances between neural processes and support the move towards more brain-based psychiatry, and thinking about how we can robustly study individual differences in large N studies. So things about thinking critically about what measures we're going to collect, trying to improve the data quality of both brain and behavior data, and using appropriate statistical and machine learning methods like cross-validation and generalization independent sample. Before I let Tyler jump up and talk about deep sampling, just acknowledgements of all the people involved in this work. Do I have any questions before Tyler takes over?
So I guess this probably isn't so much of an issue with linear regression, but with especially machine learning, very general question. How do you necessarily know what the model is coding and what you're actually learning from it? Like, especially when you use like black box, so I don't know if that's particularly what you're referencing.
Yeah, so that's a really good question. So people online, the question is, how do you, if you're training a model and you're just letting it go, how do you know what the model is learning? And that's a really good question, and not always easy to tell. So things like linear regression or things like elastic net models, these are linear combinations of features where you can look at just the weights in the features. But these more complicated models like neural nets or tree models, this is a problem because they can learn the wrong thing. There are methods that help kind of pull out like important features. So you can, for instance, you can try to retrain the model with every feature but one you care about and see how does the performance change, or things like methods like that kind of help tease apart what's an important feature in the model. But yeah, it definitely does become more of a problem with more complex features. And I will say that there has been work that looks at different kinds of models in these kind of data sets, and linear models, explainable models, do tend to do pretty well, not drastically worse than these more complicated kind of models. So the benefit of being interpretable is a nice feature, especially if we're trying to understand where the difference comes from. So. Cool. Okay. Well, I will let Tyler.
Okay. Hi, everybody. I am Tyler Morgan. I'm in the core facility, and today I'm going to be talking a bit about deep sampling, which is kind of a cryptic way of describing a certain type of study, but it fits in really, really well with what Catherine just talked about. So I'm going to talk about kind of three different aspects in my talk today. The first is just, what does that series of words actually mean. What is deep sampling? And then I'm going to go through some examples because I think the best way to get an idea of what a lot of these approaches or a lot of these studies are trying to approach is just to see what types of data they're collecting. And lastly, I'm just going to give a few brief considerations if you would like to make one of these styles of studies. So very related to what we just heard from Catherine and what we saw from the slides that she included from Dan. We often kind of have this idea in mind in fMRI, or really in any type of science that is using some kind of temporal method, that you need to balance what types of questions you want to answer with the amount of data that you are collecting and what types of data those are. Which is to say that basically there are a few different factors that matter to us when we're talking about fMRI in terms of whether we can actually see the effect that we care about. It's not so important what the math is saying here, but what I really want to point out is it's a dependence on the strength of your signal compared to the noise that you're measuring against. It also matters how big the effect size is that you're actually caring about. So some effects, like looking at a checkerboard over and over again in an fMRI scanner, they're pretty big effects. That's like one of the first effects that we ever saw in fMRI, and it's because it's huge. However, there are really, really interesting things that happen in the brain, cognitive effects, etc., that we are really, really interested in, but they are very, very small, but they still matter a lot. So we would like to be able to see those as well. Excuse me. It also matters what kind of confidence you want to have in your results. In other words, like what p-value do you care about, etc. And I have this last thing in just because it was discussed in the paper that I'm referring to, but it also matters how much time within the data that you're collecting that you can actually show a particular condition. So if your condition is something like a checkerboard, you can play it out for a very long time. That's great. There are other things like memory tasks that you really don't have the opportunity to do that. You can't just ask someone to hold something in memory forever, and you'll continue getting that contrast. So that's an important factor as well. Okay, but I also want to point out that when we talk about what noise sources we have and how much signal we have in comparison to noise, that's not just the scanner itself. Oftentimes we think about that it's like either things like thermal noise regime or physiological noise regime is a comparison that we often think about in MRI. But you also just have differences that we sometimes consider noise for our studies that are related to individual differences. Obviously, we just heard about this, so it's a pretty important factor. Many studies have tried to gain insight from looking at a whole bunch of different subjects and kind of considering them identical. This is something like a population study. Oftentimes we do this when we morph everyone's brain to look roughly the same, but we just consider that any differences that still exist are something we're going to ignore. That's like kind of what we're talking about when we show the difference between a group average of a whole bunch of different people's brains versus the detail that you can see in form and function when you're only looking at a single person's brain. So if we consider this a little bit more deeply, averaging across individuals actually results in a little bit of loss in the details that we might care about if we care about the things that are happening at the individual level. So things like just the fact that if you look at that top left image, that brain is blurrier than the one on the right. And it's because it's a whole bunch of different images overlaid on top of one another. So spatial blurring, as well as physiological differences, hemodynamic differences, etc., those are all things that oftentimes get overlooked when we're thinking about population studies. So then what is deep sampling? We can finally get to that point. It's basically just any study that collects multiple scanning sessions per subject. That's the most basic way that we can think about that. And I think we can go deeper and we can kind of make subsets of what deep sampling might look like for different types of studies. But at its simplest point, that's all it is. And as an example, hopefully you guys can see most of this. Deep sampling is aiming to avoid some of the issues that arise from these cross-subject averaging by basically looking at individual subjects, but for longer. So in other words, if we still need a lot of data to see some of the effects that we're wanting to see, but we want to avoid some of the issues that happen at the population level and the noises that are involved in that, we might just choose to have the same subject come back over and over and over again. So just to clarify, in an ideal world, if we had infinite money, infinite time, and infinite technology, we would choose to always look at this plot, which is hours of fMRI data per subject versus number of subjects that we're actually collecting. We would just always be in the top right. We would bring in thousands of subjects or the entire population. We would test them on every stimulus that we could ever think of. And then we would just understand brain activity somehow. But it turns out that because, like Catherine said, things are quite expensive, we end up most of the time being kind of smushed up against one of those axes. So in the first case, what we were talking about a little bit already is population studies. This is when you have a whole bunch of different subjects, or at least more than the standard 12 to 25 that you would have in just like a traditional fMRI study. Then you're looking at kind of population effects. And that can be that you're trying to pull apart individual differences, or it can be that you're just averaging over a lot of people because you kind of have to. Examples of this would include things like the Human Connectome project at the far end, or even what's called the Biobank project. So these are, I think the 70 Human Connectome was like 297 subjects. Biobank is, I believe, tens of thousands of subjects at this point. But those are trying to approach specific population things. On the alternative, we can think about deep sampling, which is that you have fewer subjects, but you have a lot of data. And examples of this are many. We're going to go through a few examples soon, but they include things like my Connectome, or the Midnight Scan Club, or the NSD, or at this plot actually has a marker for one of the studies, which is called the C-Neuromod study. I'm not actually going to go over it very much, but it's just, it has an insane amount of data, and not that it's just basically pointing that it's this way. And I think it's, I don't know, Dan, maybe you know a little bit better, but it's like dozens and dozens and dozens of hours for each subject. I think for one of their tasks, they just watched the entirety of Friends or something like that. Okay, so shifting into some examples, just so you can see a little bit of what we're talking about here. I'm going to start with a project from Hanke, and this is basically a really interesting data set that kind of encapsulates a lot of the things that we care about in deep sampling. And I'm just going to walk you through a little bit of what their data actually look like. So basically, the main component of their data is this center row here. And that's basically to say that they were looking at a certain type of resting state data. That is that they were listening to an audio track from a movie that all of the subjects were acquainted with, and they just listened to the whole movie. It was like 2 hours long. And then alongside that, the researchers also wanted a whole bunch of supplementary things so that they could understand these data slightly better. So first, they have descriptions and transcripts from the movie that they were watching, which was Forrest Gump, and by watching and listening. They also did a lot of demographics. They took respiratory and cardiac traces, and they also did a whole bunch of different types of anatomical scans in order to just get a better idea of what was happening in these data. And they mainly focused on modeling these data in terms of the transcripts, and then also looking at the connectivity measures within them. Kind of in a similar vein, we can think about another study, which is from a group at Berkeley. This is Alex Huth's work. And for a while, they were doing studies where people would actually just listen to podcasts, and they would listen for very long periods of time. And what this gained them was the ability to basically have individual subjects' brains individual subjects' brains be hearing a whole bunch of different types of both semantics in language and also just a whole bunch of different words. And basically what they wanted to do was then understand whether they could map those semantics onto the brain itself. And what you can see on these flat maps, or if you prefer the whole brain stuff, they're basically taking a large language model and trying to separate it into different semantic spaces. And those semantic spaces are then mapped onto different regions of the brain. And this was really exciting when it came out for, I think, for a lot of people who are doing computational work, because it basically showed that different parts of the brain were specialized to high-level concepts that we really hadn't seen in such a large scale. Okay. Just very briefly, I'm going to go back to this chart because I think an important question that we can ask is this the only dimension that really matters to us? So I'm talking about deep sampling, and basically what I'm essentially saying so far is that there are two dimensions you can have a study on. You can have lots of subjects or you can have lots of time in the scanner. But is that really all we care about? There are some other studies that actually show that the variety of tasks may also be important to that. So in other words, you can put someone in, you can just have them do the same task forever. That helps you answer a question about that particular task, but it doesn't really relate different types of cognition together necessarily. So one example that really kind of pioneered the idea that you could do a whole bunch of different things. There are actually a couple different projects like this, but I'm just going to highlight this one, is the Midnight Scan Club. For those who don't kind of know the background, the idea behind the Midnight Scan Club was essentially that it was a group of researchers who wanted to get a lot of time on the scanner so they could do a project like this. But their university had fees that were kind of prohibitory for early career researchers to be able to do that. And so they came up with a deal with their university that basically said, what if we only scan in the middle of the night? No one else is going to use the scanner at that time, but it's never off, etc. So just let us use it. And so they got discounted rates on being able to scan, but they always scanned at the same time, which was midnight, hence the name. And basically what they were trying to do was take large amounts of resting state data, which is not a new concept as we've seen so far, but combine it with the idea that there were a lot of tasks in those individual subjects as well. And they also did a lot of neuropsych batteries in order to understand more about the individual differences that they might see in connectivity and how the task contributed to that as well. Okay, moving past that third dimension that we've already added, we might also think about how much data are you really contributing to this signal noise relationship in your data. Collecting more data in a single task can be very helpful. It can increase your statistics and better understand which brain areas are related to it. But at the same time, oftentimes those types of analyses are based on models. And there are situations where we might just want to understand some aspect of the brain, but without really limiting ourselves to model-based analyses. So I think everybody knew this one was coming at some point, but a person in our lab, Javier, who you've heard lectures from already, once did a study where they basically just had people do an incredibly simple paradigm. They just looked at a flashing checkerboard and they had an attention task kind of at the same time, and they just repeated it, let's say, forever. They did 100 runs with 5 blocks each, so 500 different times looking at this checkerboard for, I think, 30 seconds at a time, just continuously, which is a terrible task to ask people to do. In case anybody was wondering, that's not a super fun task. But this actually showed something really, really important about brain activity and fMRI in general. So what they were interested in was, most of the time, if you just show someone a checkerboard, you would expect this response. You're going to see an occipital response, visual cortex response that is huge. It has a particular shape. We all think of that as the brain activity shape in fMRI, but it's not the only shape. I think we all knew this, but at the same time, it's nice to see it in data. All of these time courses from around the cortex are time-locked time courses, and we have enough power here, enough signal compared to our noise, that we can actually see what those time-locked responses look like, and that they are repeatable, but they don't look normal. Like you have on-off responses, you have negative responses, you have responses that I don't even totally understand, maybe only on, etc. And so this is a really important factor as well, and a reason why we might want more data. Okay. Next, I'm going to show what I consider kind of an extension of a project like that, which is something called the Kenshu data set. And this is basically expanding on some of the studies we've seen already where you only have audio signals, but here we're going to watch a full movie, so audio and video. And this is actually borrowing a stimulus from the Human Connectome Project's Movie 1 stimulus set. And basically they're just looking at a type of signal which is typically very, very low signal to noise. So they take Lara fMRI, specifically they're looking at vaso, and they're repeating looking at this video over and over and over again so that they can boost their SNR and try to understand whether there are patterns that can come out of this. And it turns out that yes, there are. There are also a lot of problems you run into, but that's part of the course in fMRI, especially when you're pushing kind of the limits of what the machine can do. But importantly, I think that this project really showed that deep sampling is an approach that is probably extremely kind of fundamental to the idea that when we push the scanner to its absolute limits, you probably have to accommodate that somehow. And one of the ways that you can do that is just to collect more data, basically. Okay, I'm also going to talk a little bit, well, I am talking a little bit about building better computational models that are related to this. So you rather than just repeating a single stimulus, you can repeat a stimulus that you care about different aspects. So in other words, here, rather than the people having individual differences or just repeating the same stimulus, you're actually modifying the stimulus itself to try to get at the computational units that are part of that response. This is a pretty simple form of that. So the natural scenes database, they just looked at natural images, again, kind of forever. This was over 40 scan sessions. 8 subjects looked at approximately 70,000 images. So it's one of the largest natural scenes databases to ever have existed in fMRI. Actually, it might be the largest one for this specifically. And what they care about is natural scenes kind of repeat in the things that exist in them. So they wanted to show as many possible images as they could in order to get variability in those stimuli, enough so that they could map out the responses that we see in the human brain and try to understand a little bit better what the brain is actually encoding in visual information. And it also serves a really important purpose, which is if we have these huge visual models that we're seeing through deep learning, etc., how do we compare that to the human brain? Well, we kind of have to have data sets that are approaching what these models are also built on. And so that's an important factor, is being able to have comparative data sets in an encoder like the human brain versus an encoder like a machine. Okay, there's one last section of examples that I'm going to show, and then I'll be pretty close to done. But I also want to talk a little bit about, you might want to look at changes to the same brain over time. So as one example of this, I'm going to talk about the My Connectome project. This is actually just one individual. This entire data set is just one person. He was scanned over the course of a year approximately 100 times. And importantly, they collected a lot of different data to try to understand and explain why resting state data and connectivity measures might be changing over that time in that individual. So they collected the resting state data, but they also included daily surveys. They also looked at blood draws, and they even collected genetics, et cetera, to see what types of things were specific to this individual and what types of things changed over time. I think it's a very cool design. It also kind of gets into the idea of what we talked about, or what Catherine talked about with individual differences. But here, the individual differences are not individual people, they're days of the same person. Another example that I think is really amazing in this domain is the idea that our brains are never really the same. And that can be related, like we talked about, with different things that are happening, but some of those are very easily measurable. So in this case, Taylor and colleagues looked at hormone levels in the blood over the menstrual cycle and actually were able to find it wasn't just a functional change, it was a physical change in the hippocampus that was happening over that cycle. And so then they were able to take, in this particular case, they scanned someone for every single day during their cycle. And then they were able to actually look at blood draws and hormone levels and relate those to the volume of different hippocampal subfields. And what they found was that progesterone was related to the actual volume of a number of different areas in the hippocampus. This is cool, but also what I think is maybe even cooler than this is that then they scanned the exact same person. They repeated the entire experiment, but on birth control, in order to show that when you reduce progesterone entirely, that those fluctuations actually diminish quite a lot, which kind of indicates that it's like an individual subject modification of their hormone levels in order to determine how that is affecting their own body. Okay. Lastly, in the last 3 minutes, I'm going to talk a little bit about the considerations that you might want to keep in mind if you're ever thinking about a study like this. So there's a really nice paper that came out relatively recently from Cooper's, and they actually define 4 principles that are really important if you're going to have a large study like this. The first is that, and this might seem obvious, but you should design it well. You should have experiments that are motivated by theory and they elicit specific cognitive processes that you care about. Statistical power should be maximized and protocols should ensure that participants actually do the task. As someone who has worked in this field previously, particularly with data that's quite noisy, if you find out at the end of the 5th or the 10th session of a person coming out of the scanner that they weren't actually doing the task, that sucks. It wasted your time, it wasted their time. Well, they're still getting paid, but you know, it's not great. Okay, the second thing, and again, is probably obvious given what I've been talking about, is that you should scan more. You should get a lot of data, but you should also keep in mind that scanning more doesn't always just mean the task that you care about. You can also care a lot about different anatomical measures, things like T1 weighted and T2 weighted anatomicals, venograms, angiograms, diffusion weighted MRI, quantitative MRI. There are a whole bunch of different relationships that you may not think of initially, but others would be really interested in understanding in comparison to the data that you cared about. You should also make sure that your quality is high. So if you optimize quality, this can include things like experimental protocols, making sure that different people who are collecting data are doing it in the exact same way, making sure the timing is allowing you to analyze those data correctly, and making sure that just the SNR of the scans that you're looking at, the image quality and the participant monitoring are all working properly all the time. And it can also be really important to think about how you're going to process these data, because these data should be relatively uniform, but there are also things that are going to come up every time that are pretty important to keep in mind. How is your pre-processing going to change over time if something happened during the scan, or even just in general, how are you matching to what the community is doing, etc. Things like different types of filtering, blah, blah, blah. And also lastly, it's really important to think about if you're putting a person in multiple times, things can go wrong. So how are you going to make sure that your cross-session alignment is really, really good? Lastly, and this is not something I've talked about yet, you should think about how you can share those data effectively. It's we as a community are thinking about a lot of the same problems. And some of the data that we have, if they're extensive enough, can be used by others in order to answer really important questions that, again, you may never have thought about. So making sure that the data that you do collect is curated really well, it's documented really well, and it's accessible to a lot of different people is really important. And especially given we are at NIH, that's part of our mission, is in order to make it so that different researchers have the opportunity to ask important questions. Okay, this is my last slide. I want to leave you with a point, if you are thinking about studies like this, that the kind of space of questions that we can ask in fMRI is quite grand. It's huge. There are a whole bunch of different things about the brain in particular that we can try to address. We can ask questions about visual cortex or motor cortex or memory, etc. And those are all different inferences in the scientific space. But importantly, what our goal is typically in deep sampling is to try to cover more than just one little aspect of that space. We want to cover as many hypotheses as we can, and that's probably how we want to design them. Lastly, it's also helpful if you know of other data sets that are deep scanning or deep sampling or intensive, whatever you want to call it, that maybe there's a little bit of overlap with those, because those can kind of link the spaces within scientific inference in a way that gives us progress faster in the field. So I'll leave you with that. And if there are any questions, I'm happy to answer them. Cool. Okay. And Catherine and I are both also available either just by stopping by, or if you want to send us an email if you have questions that are specific or you think of later. And in both of our cases, our emails are just our names followed by NIH. Okay, thank you so much.